Welcome back. Now we are going to take the individual antimicrobial compounds and discuss their chemistry and biochemistry. We will just recapitulate. We start with recapitulating these compounds, which I think we have already discussed. This is the structure of trimethoprim, an antimalarial compound. This is a sulfone, para-amino phenyl sulfone, that is an anti-leprosy compound. This is sulfomethoxazole, which is an antibacterial compound. You see here it is written bacteriostatic. That means, these are antibacterial agents which inhibit the cell growth. Why these are bacteriostatic? Because these are the compounds which stops the folic acid biosynthesis. Okay. Folic acid biosynthesis, which is a one carbon transfer, remember, it is nothing but conversion, a very important conversion, that is uracil to thymine, and that was ultimately that is the consequence of perturbing the folic acid biosynthesis. So, one of these compound is like sulfomethoxazole or this anti-leprosy compounds. This is little bit different because that inhibits the DHFR of microorganisms, okay, DHFR, dihydrofolate reductase, and these two are nothing but you can write them as SO2 NR. So, para-amino SO2 and then NR. We have told them at that, I told you at that time that it mimics the para-amino benzoic acid, which is a component of the folic acid. Okay. So, para-amino benzoic acid has to be incorporated into a pyridine nucleus, which is a dihydropyridine nucleus. If you remember, go back to your earlier notes, it is the 7,8 dihydropyridine that is formed. So, that reacts with this para-amino benzoic acid, and the enzyme that does that is called a pteroid synthase. Now, this pteroid synthase must be such that there is a hydrogen bond, it goes to the active site of the enzyme, and this is the NH2, which forms a hydrogen bond with some amino acid here. There is a aromatic amino acid somewhere here, it must be, so that there is pi stacking interaction with this benzene ring, and then there must be some ionic interactions. That means, there must be some sites like NH3 plus, which forms an electrostatic interaction here. So, the para-amino benzene sulfonamide, that you see the similar structure. So, NH2 is there, that forms this hydrogen bond, this is the aromatic ring, and this is, instead of the CO2 minus, you have SO2 minus. So, that forms the ionic bond. So, this is a very good competitive inhibitor of para-amino benzoic acid. These compounds are also called anti-metabolite. What is anti-metabolite? Which interferes with the metabolic processes that are going inside the microorganism or anything, any species. Okay. And this, since this is now inhibiting the incorporation of para-amino benzoic acid, that means it disturbs the normal metabolic process. So, this is a very good, initially these are very good antibiotics, but today the bacteria has developed resistance against the sulfonamides. How? By producing more of para-amino benzoic acid, and you know that in competitive inhibition, if you increase the concentration of the substrate, you can overcome the inhibition. That is the nature of competitive inhibition. So, now sulfonamides are virtually not working against any microbial infection. Now, let us come to the chemistry of penicillin. So, this is the structure of penicillin. What is the pharmacophore of penicillin? Now, pharmacophore is the entity, molecular entity that is present in the entire molecule. Somewhere it is present, and that is essential, or which that part is interacting with the with some enzyme or any nucleic acid, whatever is the target. So, basically what we are saying, that if I have a big molecule like this, and if I see that only this part is the one which is interacting with the enzyme, then this part is what is called the pharmacophore. Okay. So, the pharmacophore of penicillin is is this which is put as light green, okay, a light bluish. So, that part is the pharmacophore. Now, this part has what is there? There are two rings which are fused to one another. Okay. Now, this is called a beta-lactam ring and this is called a thiazolidine ring. That is the name of the heterocycle. This is beta-lactam fused to a thiazole, okay. And then there is a substituent amine here which is acylated. So, you have an acyl side chain here and you have a carboxy also, alpha carboxy group attached to the thiazolidine ring. Okay. Now, this is the minimum. So, any penicillin, the variation that we see in different penicillins, you know that the names of different penicillins, ampicillin, amoxicillin, cloxacillin, methicillin, whatever, name any penicillin, the difference is only in this R group. So, you have different R groups and you get different types of penicillins. Okay. Initial penicillin that was made was benzyl penicillin called penicillin G, and another one was called penicillin V, that was phenoxymethyl penicillin, phenoxymethyl penicillin also called penicillin V. In fact, penicillin G is still available in the market. It is usually now treated for rheumatoid arthritis. That is usually the compound, this benzyl penicillin that is used. Otherwise, for other bacterial infections, a respiratory infection, usually this penicillin G is not used, but historically these two compounds are important, because the first penicillins that were introduced into the market were this benzyl penicillin and this penicillin V, penicillin G and penicillin V. Okay. This is what is the we will talk about this biosynthesis later on, because we know that penicillin is made by the microorganism, that is why this is an antibiotic. Okay. Now, that means the microorganism has a biosynthetic machinery to make these penicillin enzyme systems, which starts from a building block and join them together and finally get to the penicillin. We will discuss this formation of penicillin in a much elaborate way, but here just to let you know that it is a very simple, these three simple building blocks which ultimately combine and make the penicillin. One is, this is a variable amino acid, this is cysteine and this is valine. So, cysteine, valine and another amino acid which I am not naming it, actually this amino acid is alpha amino, this is not a protein amino acid, this is alpha amino adipic acid. Alpha amino adipic acid. So, basically this alpha amino adipic acid and through this alpha amino adipic acid combined with cysteine and that also combines with valine to make what will be a tripeptide, what will be called a tripeptide, and that tripeptide then cyclizes and you have this penicillin molecule. Remember the alpha amino adipic acid, it is the carboxy, that is the epsilon carboxylic acid that reacts with cysteine and cysteine reacts with valine to make a tripeptide which will be called ACV, adipoyl cysteanyl valine, that cyclizes to give the penicillin. Okay. Now, another important interesting aspect of the structure of penicillin for which the activity is there is the reactivity of this beta-lactam ring. Reactivity. Reactivity to what? Reactivity towards nucleophile, because amide carbonyls are susceptible to ring opening, okay, to hydrolysis. Like if you take an amide RCONH2 and if you heat it with alkali, you get RCO2H or RCO2Na, because you are using alkali. say NaOH, and you get ammonia, and if it is NHR, then you will get RNH2, the amine. Okay. But in order to do this, you have to heat it with alkali. It is not a very, means it does not work at room temperature, or if you want to do it at room temperature, then you have to wait for months and months, so that the hydrolysis will take place. Okay. Now, why amides are difficult to hydrolyze? Because of this resonance. The nitrogen lone pair is resonating with the carbonyl. So, you have a resonating structure where the nitrogen is planar, and this bond is also becoming very rigid. So, that will not allow the things to rotate. This is exactly what happens in peptide bonds. When we discuss the structure of peptides, we have seen that the amide bond is this nitrogen carbon has a significant amount of double bond character, and that stops the rotation. Okay. So, this is the normal amides, and because of this resonance, this carbon is losing the electrophilicity, because if there is only carbonyl, then the oxygen will pull the electrons towards itself by electromagnetic effect, and then this will have a positive charge. But here nitrogen lone pair is there, that will neutralize the positive charge on the carbon. So, nucleophilic attack on the amide is or requires higher temperature, okay, or higher activation energy is required. On the other hand, in penicillin what happens? This is the structure of penicillin. See, it is not a planar molecule, it is like this is a planar beta-lactam ring, a 4-membered ring is almost nearly planar, and then you have this 5-membered ring which is going upwards. So, basically it is like a open book type. So, basically something like this. So, it is something like this. So, this is the 5-membered ring, sulfur, this is the carbonyl, this is the nitrogen. So, it is like a open book type of, or a butterfly type of structure. Okay. So, it is not a planar molecule, that is number 1. Number 2 is this, whatever in the amide, in acyclic amides, this lone pair is entering into resonance with the carbonyl, that is actually not permissible in beta-lactam, because if the lone pair participates in resonance, then what happens? The angle becomes 120. That means, the angle strain in a 4-membered ring, the angle is virtually 90. So, that means, you have already deviated the angle quite much from 90, you are deviating is 90, but actually the hybridization demands that these have to be 109.5 degree. Okay. So, if you have a resonance where the angle is still increasing further here, so that will not be allowed. So, this will be flat and it will be highly strained. So, this type of resonance is almost not possible, and that is reflected if you take the IR spectroscopy of penicillin, then you will see that what happens in IR spectroscopy, if there is resonance, then the carbonyl stretching frequency will be low, and if there is no resonance, the carbonyl stretching frequency will be higher. So, in penicillin, the carbonyl stretching frequency is about 1770 centimeter inverse. On the other hand, in the amide carbonyl, it is 1680, around that value. So, that shows that it is a very highly strained molecule, and the carbonyl carbon is the carbonyl is virtually present as a separate carbonyl. There is no such assistance from the nitrogen lone pair adjacent. So, that makes its carbon highly electrophilic, that means, that makes its carbon highly susceptible to nucleophilic attack. So, that is the basic that is the one of the major reason why this molecule acts as an antibacterial agent. Okay. We will slowly go back to that, but again before I go into any further, I should tell you that whatever mechanism that we are discussing today, that this is the way the penicillins work, that is what is called post facto analysis. That means, it is after the fact after the fact that penicillin is a very good antibiotic. It kills the microbes, it kills the bacteria, and once that is known, at that time Fleming or Florey or Chain or Dorothy Hodgkins or even Woodward, Robert Robinson, they did not know how it is working. Only thing they know that it is a very good antibacterial agent. Okay. Of course, that ultimately matters, because ultimately it matters. We don't some people might say that I do not care how does it work, but it is working nicely to cure me of the infection. Okay. But however, science can stop there, science has to progress. We have to know how penicillins work, because until we know that, we cannot devise or rationally design new antibiotics based on the mechanism of action of penicillin, because today we are living in an era where most of the earlier penicillins are not working. So, now how to make new penicillins, new type of antibiotics, that depends how is our understanding of the antibacterial activity of penicillin. Okay. So, that is why, so just to make it very clear that whatever we are discussing now, this is after the discovery of penicillin, after the penicillin entered into the market. So, the mechanism was established. Okay. Now, what is the mechanism? I told you one thing, that penicillins work against the bacterial cell wall. Okay. So, we have to now know what is a cell wall in a bacteria, that is so unique, for them. Okay. Cell wall, I told you, it is a rigid wall that protects the cytosolic material inside the bacteria. Now, there are two kinds of bacteria, one is gram positive, another is gram negative. Okay. So, gram positive bacteria has a thick cell wall structure, and it is written peptidoglycan. What is a peptidoglycan? Peptidoglycan means you have peptide units as well as glycan units. Glycan means sugar units. Okay. So, basically you have carbohydrate, which is the part of the sugar, and you have peptide. So, that is why this is called peptidoglycan. So, the one thing, that the cell wall is made up of peptidoglycan, that is number one. Number two is that in gram positive you have the peptidoglycan as the outer wall, and then you have the membrane, like we have this lipid bilayer membrane, and in that membrane there will be definitely channels, because bacteria has to communicate with the outside world. Some molecules will come inside the there are carrier molecules which will carry the outside whatever is needed. So, this is the cytosol on this side, cytosol. So, some molecules are needed, water is, so it has to be controlled, like ion channels I told you. So, ion channels, ligand gated ion channels, or these things. So, bacteria has these pores also. So, through that pores materials enter or go out. Okay. So, this is your peptidoglycan, and that is quite thick in gram positive bacteria. In gram negative bacteria, what happens? First, it is an outer membrane, which is a lipid bilayer, and then there is this pores. These are called porine channels. Porine channels. Okay. Then there is a much thinner peptidoglycan. That was earlier for the gram positive, it is outside. Then little bit inner, that means this is protected by a lipid bilayer membrane, and then you have another membrane, an inner membrane, and then the cytosol. So, there is a distinct difference, and you can now say that penicillins work against the gram positive very well, because in the gram positive the peptidoglycan is exposed outside. So, penicillin comes and destroys the peptidoglycan, but here the penicillin has to traverse through this. First, this is the barrier. So, gram negative bacteria then come here. These spaces are, by the way, called periplasmic spaces, and these spaces where the peptidoglycan is there. So, then it has to come inside here and then act on the synthesis of the peptidoglycan. So, it will be difficult to kill the gram negative bacteria by this type of mechanism. But anyway, let us see what are the other problems in gram negative bacteria. Gram negative bacteria has other sorts of problem. So, we will discuss that. What is this? Let us inspect it at the molecular level, the peptidoglycan. Okay. Here this is the schematic diagram. You see this is one sugar unit, that is called NAM. What is NAM? That is, NAM is N-acetyl glucosamine, and then you have another sugar unit which is called NAG. This is called N-acetyl, NAM is N-acetyl, extremely sorry, that is actually N-acetyl muramic acid, NAM. This M, this A and this A, and the other is called NAG. NAG is N-acetyl glucosamine. N-acetyl glucosamine. So, these are the two sugar units, they are joined one after another, NAM then NAG via glycoside linkages, NAM NAG, NAM NAG, NAM NAG. So, this way some chains of sugar is there, then another chain of sugar is there, then another chain of sugar is there, and each sugar unit, not each sugar, in the each strand of sugar polymer, what you see that in the NAM, N-acetyl muramic acid, what happens that there is a peptide which is attached certainly to one of the hydroxy. So, that is attached, the first amino acid is L-alanine, then D-glutamic acid, then L-lysine, then D-alanine, and actually there was another D-alanine, but by reaction while forming this cell wall, one of the D-alanine goes out. So, basically we are just we are not discussing the formation of the actually formed, this is the structure of the when the matured cell wall is there, but we are discussing just before that, before the formation of the matured cell wall, what is the status? The status is like this, that it one of these, that all these peptide bonds are there or amino acids are there. So, this is what a pentapeptide 1, 2, 3, 4, another one before the formation of the matured, remember the matured cell wall. We are talking about just before that, what was the status? There was 5 amino acids, they are hanging from one strand, and in adjacent strand there is again the 5 amino acids are hanging from each other. Okay. Now, each peptide has a side chain also, has a side arm, which is reactive arm, like this one has a side arm. Then there is another one, suppose here, which has got a side arm, and then there this has got a reactive side arm. Now, there will be a reaction between this strand and the side arm. So, there will be a attachment like this, then there will be an attachment like this. See, if that happens, then what happens, the cell becomes very rigid. It is like that if you have lot of bamboo rods and then you have put it on to make a fence. If you want to do that, only putting these bamboo rods, the bamboo poles will not be sufficient to protect. So, in order to make them very rigid, so what you do? You do this type of what are called cross links to make it very strong. When you build a house, that is the same thing, beam and pillar. So, beam and then pillar and then you join with the horizontal pillars. Okay. That is to reinforce the structure. So, to reinforce the structure, this cross link is required. Okay. So, basically now what we have learned, that there is this glycan units, glycan polymer, that is NAM, NAG, NAM, NAG and NAM, NAG. Each of these NAM is attached to a pentapeptide, and then there is a cross link between the adjacent pentapeptides. Okay. So, we will look at very carefully what are these reactions, how this cross link is formed. Here it is now written in a little bit better way, that N-acetyl muramic acid, that is basically this one. Let me write the structure of the sugar. So, this is the glycoside linkage. This is glucosamine. N-acetyl glucosamine means NH. Glucosamine means is glucose, only the 2-hydroxy is replaced by an amine which is acetylated. So, then you know in glucose what happens, this is alpha, then the next one is beta, the third one is, this is number 1, this is number 2, this is third, this is the number 4 carbon, which is alpha, and then you have the CH2OH. So, when the glycoside is made, it is made between 1 and 4, that means this is attached to another sugar. Okay. So, this is your NAG, N-acetyl glucosamine. And what is your NAM? The NAM is like this. NHAC, N-acetyl, that is there, and this is attached to the glycoside, another sugar unit, and here the weight is beta. So, that is attached to a lactic acid like this, and this is CH2OH. So, this is, see what is lactic acid? CH3CO2H OH. So, it is attached by a ethereal linkage, ethyl linkage. Okay. So, this is your NAG, this is your NAM, and this is your NAG. So, this is how it is done. So, this is NAG attached to a NAM. NAM has a carboxy which is attached via a lactate side chain, and this carboxy is the one which is used to make the peptide. So, it will be CO, then there will be alanine, then there will be glutamic acid, then there will be lysine, then there will be alanine, Okay. So, I hope this is clear. Now, that will be, we are not writing the NAM NAG, just we will write the structure, we will write the NAM. So, NAM, every NAM is attached to alanine, glutamic acid, lysine, alanine. Okay. Also, look at the configuration of this alanine. This is L-alanine, this is D-glutamic acid. I told you that in bacteria only that there are the D amino acids are present. Where from these D amino acids come? By isomerization of the L amino acids. Okay. So, then L-lysine, but this lysine, the side chain amine is attached to glycine, 5 glycine units. Okay. So, what is the end point here? Is it the N terminus or C terminus? That we have to be clear now. See, this is your lysine ends up with NH, then you have a glycine. So, NHCO glycine, then glycine ends up with NH2, that is attached to another glycine. So, NHCO. So, if that be the case, you can say that it ends up with an amine. Okay. That is very clear. If somebody says this is amine, but you have to make yourself convinced, yes, because lysine starts with NH. So, NHCO, then ultimately does not end up with a CO2H, it ends up with a NH2, and then you have D-alanine, and in the adjacent NAM-NAG you have the same chain, alanine, D-glutamic acid, lysine, that has got pentaglycine, D-alanine. Okay. Now, the reaction that takes place in cross linking is, I told you, this side chain reacts with the primary chain that is hanging here. Now, this D-alanine, again you have to be sure that what ends up here. You started with NAM is this one. So, this is CO, then NH. So, alanine NH is on this side and CO2 is on this side, that means this will end up with a CO2H. So, this is a carboxy end here, okay. And in between D-alanine you have a CO-NH. Okay. So, you have a D-alanine here, you have a D-alanine here, that is the end point, and this is the CO2H, and this D-alanine is attached to lysine and all these things. So, now the reaction that takes place is basically that this NH2, that the glycine NH2 attacks this bond, this amide bond, okay, and kicks out the last D-alanine. So, what will happen now? This is the chain that is now cross linked with this chain, and the result is nothing but, I can simplify it. Suppose this is glycine that has got NH2, and then you have D-alanine. So, D-alanine, then CO, then NH, then D-alanine, and this was going to the top attached to an M, and now the reaction is basically this is attacking here, that goes out, that makes a cross link. But the reaction is, what is the nature of this reaction? How can you classify this reaction? This is nothing but, look at this D-alanine, this was connected by another amino acid D-alanine earlier. Now, what you are doing? You are replacing this D-alanine and replaced by what is glycine. So, this can be called a transamination, a transpeptidation reaction. It is not transamination, amine is only amine NH, but this is called transpeptidation. That means, your peptide was having another amino acid that is replaced by a new amino acid. Okay. So, what is transpeptidation? That if you have a peptide like this, and if you have another amino acid, and if you get AC plus B, that is a transpeptidation. These are all amino acids. Okay. So, this peptide bond is replaced by a new peptide bond. So, that is why this is called transpeptidase. So, there is an enzyme to do this, otherwise this reaction will not take place. This enzyme is a serine based enzyme. So, there is an enzyme, it is written here, enzyme OH. So, initially enzyme OH, like chymotrypsin, what happens? When you hydrolyze a peptide bond, you attack the peptide bond with the serine OH, replace, at that time you break that other amino acid, and then this serine is released again when the water comes and attacks. Okay. In this case, not water, the enzyme attacks the carbonyl, forming the tetrahedral intermediate, the D-alanine leaves, and then instead of water, now the adjacent glycine amine attacks and releases the serine OH. Okay. I can show it again even better way, that basically what we are talking about, CONH, D-alanine, and this is D-alanine, and on this side there is this glycine NH2. So, initially the enzyme which is having a serine OH that attacks here forms the tetrahedral intermediate. So, D-alanine, CO minus, and then NH, then D-alanine, and this is the serine enzyme. Now, what will happen? So, there must be some oxyanion hole here which will stabilize this O minus. Now, what will happen? This comes back, that goes out. So, D-alanine has gone out. So, D-alanine and then CO, and that is attached to the serine, and now the glycine which was waiting for this to happen comes, attacks, breaks this bond. So, you get the link between the glycine and the D-alanine. So, now the biosynthesis of cell wall, what happens here? Look at the workload on the bacteria. The workload on the bacteria is that it has to synthesize the NAM, it has to synthesize the NAG, and then combine NAM and NAG, form the glycoside bond and make the polymer, make another polymer set of polymer, another set. So, all these rods which are made up of, or layers which are made up of NAM, NAG, NAM, NAG, and then it has to also do this muramic acid, N-acetyl muramic acid, that is also it has to make and then form the peptide, all these. And this is the last reaction in the synthesis of the bacterial cell wall. This is the last reaction. So, the last reaction where this transpeptidation reaction, that is somehow stopped by penicillin. So, how penicillin works? It stops the enzyme transpeptidase from working against. But it is a beautiful way to really harass the bacteria, because you are not killing the bacteria or stopping the bacteria from the very beginning. You have done enormous amount of work, you have prepared for an exam reading 24 hours a day, and on the final day you could not answer anything because the questions are so hard. Okay. So, basically that is the case. The bacteria has worked to make all the cell walls, all the components assembled together. The last one, okay, the last one, like in the marriage ceremony, if the priest does not arrive, the ceremony cannot take place. So, it is basically that at the terminal point you are stopping the bacteria from forming the cell wall, and if that cross link does not happen, the cell wall is very fragile. Water will now go through the cell wall inside the cell, and then the cell burst open, and that is what is called lysis. Now, the question is, why penicillin will stop this? Will stop this cell wall formation at the terminal stage. What is the similarity of the reaction or the structure? Okay. But before that, I may mention that this the structure that I gave you, that lysine, then glycine, 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 this is usually present in gram positive bacteria. In gram negative bacteria like E. coli, this lysine is replaced by an amino acid which is called diaminopimelic acid. Diaminopimelic acid. Pimelic acid is, by the way, what is pimelic acid? Oxalic acid, malonic acid, Ohm's gap, if you remember, P is the C7. So, you have CO2H, NH2, and then all this, and then another NH2 and CO2H. So, this diaminopimelic acid you might see in different books that this is diaminopimelic acid, but the mechanism is same. In that case, the amine side chain of the diaminopimelic acid will react with that. And incidentally, this is the mesodiaminopimelic acid, because this has got two stereogenic centers. If they are of opposite configuration, that becomes meso. Okay. It is the mesodiaminopimelic acid in E. coli that does this job. Otherwise, in gram positive, what we have discussed is gram positive bacteria. The glycine is the one. This is normally written in the books, in the medicinal chemistry books, but the mechanism remains the same. Now, let us see what is it is there, mesodiaminopimelic acid or lysine, any one of these, but lysine, then that pentaglycine has to be attached. It is through the glycine side chain. This is the mechanism which I have already discussed. Now, let us see. Penicillin is an inhibitor of this transpeptidase cross linking reaction. Okay. Transpeptidase reaction takes place if this is the transpeptidase enzyme, then the this is the D-alanine. Remember, this is D. So, that has to be configuration of R. So, 1, 2, 3. So, that is R, and this is also 1, 2, 3, but the methyl is alpha. So, this is also R. So, this is the D-alanine. So, what is the reaction? The serine which, remember why this D-alanine is binding to the enzyme, there must be a positively charged species which is like lysine. So, that forms the salt bridge. So, this goes and binds, and then the serine which attacks forms the acyl enzyme complex, and from the other peptide chain which is the glycine one, now the glycine NH2 attacks and forms the, this is the cross link. Okay. In case of penicillin, it has been found that instead of D-alanine, penicillin comes, the bacteria thinks that penicillin is my substrate, and penicillin comes, and the serine, it has got this CO2 minus that binds with the lysine, and this which now this is a very reactive ring which can open up immediately. So, which attacks, and via the formation of the tetrahedral intermediate, it goes back and kicks out this. So, this is the situation now. So, the serine is acylated by the penicillin, and what happens now, the serine can be regenerated provided water comes and breaks this bond. So, that the enzyme is freed again, but this is a very slow reaction. This is a very slow reaction. If it is a very slow reaction, by the time this hydrolysis happens, the bacterial cell is already lysed. Okay. Bacterial cell will not wait for such time that the enzyme is free again. So, basically essentially it is an active site directed irreversible inhibition, active site directed irreversible inhibition. What is active site directed? That it goes straight away and attacks the enzyme. That what happens? It attacks the enzyme and stays there. Formation of covalent bond means it is a irreversible enzyme, and then after the reaction nothing, no more further, any further reaction has not happened to produce a more reactive species, that is not happened. So, this is nothing but an active site directed irreversible inhibition, ASDII. Okay. So, ASDII. Okay. So, this is the mechanism, this is written here. Water attacking this is blocked, water cannot come and hydrolyze this one. Okay. So, that is the mechanism of penicillin. Now, the last thing that is remaining is why penicillin is so selective, why bacteria thinks that this is my substrate. Now, compare the two structures. This is the structure, again another way of writing D-alanine. See, this is the methyl, you see this has to be R configuration 1, 2, 3, because the methyl is alpha. So, it has to be R now. So, that is D, and this is also D. So, I have written this structure, the D-alanine in this fashion, and look at the structure of penicillin. See, this is same. If you look at the backbone only, you will see the similarity of penicillin and D-alanine. Okay. So, that means penicillin mimics is only C, I think, no? mimics D-alanine in structure. It is only DA, it is not LL. If it is LL, bacteria had used LL, penicillin would have been ineffective. The stereochemistry as well as this stereochemistry is also matching. Only thing, penicillin has some extra is that this part is what is the extra. So, it is even better, it is a conformationally constrained system which resembles D-alanine. Of course, D-alanine must be having different types of conformation, but in one conformation you see that it resembles the penicillin. So, now in short, what is the mechanism of action of penicillin? It stops, it is a transpeptidase inhibitor. What is transpeptidase? Transpeptidase is a indispensable enzyme to make cell wall. What it does? The terminal step, again I repeat that the bacteria has made the cell wall, premature cell wall. It is like making premature or immature mRNA, and then if suppose you stop now the splicing with the spliceosome. So, your mRNA will be ineffective. So, similarly, it makes the everything except the cross linking. So, at the cross linking, terminal cross linking, that is inhibited, and that is how penicillin works. Okay. I think that is all for today. For this session, we will talk about the bacterial resistance in the next session. Thank you.